So please, this is our opportunity to talk about the issues around the technical measurement and our understanding of tumor heterogeneity, and not just the technical questions from the talks, but you have an opportunity to quiz some of the world leaders in addressing these issues.
So I was having a little trouble grasping the scope of what's possible with single-cell sequencing. There was one discussion of 1 million cells for $1,000. How much can we do, and how? Okay, 1 million cells for $1,000. How much can we do, and how much does that relate to being able to characterize a macroscopic bulk tumor? I mean, are we a 0.5% of the way there, or 80% of the way there in terms of practical terms?
So this is really a flow cytometry experiment, and the costs are associated with the antibodies. So, to do about 100,000 to 1 million cells, it is really about actually $500 to $600 for the antibodies themselves and the reagents. The other amount of time is basically how long it takes to run the sample.
And so what is 1 million cells? I'm not familiar with the number of cells in a macroscopic tumor. What percentage of cells are we talking about there then?
Well, I mean, well, the tumor they took out of me was about that big.
Right.
So, I mean, that's 1 million cells is, less than the bottom of an Eppendorf tube. So, I mean, I think that comes to the question that the gentleman over here asked about, how much are you sampling? It's a sampling error problem. I mean, if it's a solid tumor, unlike a liquid tumor, liquid just randomizes. Solid tumors are basically branching structures. Think of it as a complex, coral tree. And so if you could sample along the tumor mass, you might actually be able to pick up, in fact, some of the descendancy of the genetics.
But it would seem like the key strategy then is to combine some of these single cell samples with some of the more macroscopic and draw
well,
a more
that's what the
complete picture.
MIDI approach was meant to start to address. We've set it up, and the design is so that we can get a 1 centimeter square in basically about an hour or so. That's the objective of the new instrument that we're building. So, like with sequencing costs, it's all about time and money. And, I think, though, that what I would look at is, I'd say the phenotyping, where we're able to basically take advantage of 30 years of flow cytometry. I see the day, definitely, when the genetics will be cost effective, where you can start to think about 500 to 1 million cells. It's not today, it's not tomorrow, but it's going to come. Sequencing is going to, even as tags, is going to replace fluorophores.
I think this gets to a related issue that's been addressed a little bit about I think of it as the phylogeography of the tumors. What's the relationship between space and the heterogeneity? And it seems to me, and please chime in, but it seems to me there's an open question: to what extent is there cell migration that's mixing the clones? To what extent are they really just dividing locally and causing little uniform patches? But I believe that's an open question in solid tumors.
I agree.
So I guess I had a question exactly on this point, I guess particularly for David. In particular, if there only seem to be only a few types you've identified within the tumor, and what are the prospects for putting on a tag pre-sequencing which has spatial information in it? Then I would have thought that you have big enough regions and enough data from that, even at 50x coverage, that you might be able to get substantial spatial information from that, at least in principle.
It would be very interesting to look at tagging different regions within the genome before you do whole genome sequencing.
I want to tag different spatial places within the genome. Spatial places,
that's
right. So instead of grinding it up first.
That would be great. Whether, that's the approach, or more like Steve is doing, where you pick out individual cells as exemplars of the different parts of the geometry of the tumor. I don't know. I mean, it's a great. We'll see. What's your opinion on tagging and then whole genome sequencing?
Well, it's
single cell.
I mean, there's this literature where people will dissect the tumor and prepare the libraries separately from different regions. Right. So you don't need to tag it so much as just slice and dice before you grind.
Right.
Right, there you go.
Charlie Swanton will be talking about it tomorrow. Charlie Swanton will be talking about that tomorrow.
Okay.
But even with 50x and total coverage, even if you have many more than, many more than 50 regions, which you're doing that with, you still can get a lot of information because of these regions and everything are long enough. And
it's more informative than mixed all together, grind it up. And
I think there's a continuum to the trade-off as well, as far as number of regions versus depth per region. Just like with single cells, the more information per cell versus number of cells. cells. I mean, you can do a whole genome from a relatively small number of cells, and that doesn't tell you a lot because you haven't sampled so much.
Right.
Or you
Right.
can look at a small part of the genome and a lot of cells, and
Yep.
those trade-offs are at the heart of
Yeah.
sort of all the experimental design here, I think.
But it's going to depend a lot upon these questions about whether the heterogeneity is a lot coming from migration or things arising in different regions and sort of reasonably big, compact regions which are different types, because the kind of information you'll get and so on.
But I think part of that goes
That'll
back
be
to the
the
very
difference.
first talk where, Nellie's making measurements in situ. And so again, at very targeted questions, you can make in situ measurements that retains, at the level of the individual cells, their spatial relationship. And that's very useful as well.
Mina?
I'm here to cause a little bit of controversy so that we can have a more, what's the word? A more difficult discussion. You three are amazingly talented, and you make these gorgeous, Gary, be quiet. So,
Somebody turn off her mic now after she said
Hey, stop right there. That's very good. Let's move on.
Was that the
So I am-
controversial part?
so I'm going to basically actually say something about what David said. So David started with this very exciting, lively discussion about how tumors are not going to get any smarter, and we are so smart, and once we figure it out, then that's it. So I like to propose that that's a little bit. What's the word? I don't want to say anything that is, anyway. So what I would like to say is that I would like to challenge that question because, that's a statement, because I think tumors are basically evolutionary organs. And that, in fact, I will show that at the end of my presentation. This is what I think, and I think it has a lot to do with aging. So I would like to propose that we are not going to eradicate cancer. Tumors are going to be with us as long as aging is with us. And until such time that you actually understand aging, you're not going to be able to get rid of tumors. They're going to evolve with every individual depending on how they are aging. So there are fundamental things about tumors to understand. They are actually quite clever. And they evolve in the most amazing way. They are not dumb at all, as Nellie's beautiful talk showed, and as many of your talks shows. So I think it would be good to think about tumors in these lights, and while we are developing these magnificent technology, to try and really understand the biology. And the biology is going to be a lot more complicated than defining the cell types within a tumor or defining single cell, whatever it is they are doing. So I think in that light, it would be good to combine these things with models and thinking that actually reflect what goes on within an individual and how they relate to aging. I guess I'm putting a plug for former postdocs who work on aging, and I have pushed them in that direction because I think it's a very important area in relation to cancer. So that's my little bit of troublemaking.
Well, I thought Calico was going to solve all that for us. And,
It's what?
I thought Calico was going to spend $1 billion to solve that problem for us, precisely
Oh,
that
yeah?
problem.
I like David to make a comment about how stupid or how smart tumors are.
Let me completely clarify what I was saying. Tumors are very smart, all right? They're smart. All I was saying was that your child's tumor won't be any smarter, right? It was strictly a comparison. And that's not true for infectious diseases, right? They pass on their DNA to future generations, and they can pass on strategies for evading our therapies. But cancers don't pass on the strategies that they have for evading our therapies to the cancers that our children will get. This is a fact. Right? That's everybody says, "Oh, you make it you make it sound so easy, and we're going to cure cancer and so forth. But if you take the long run of this, we'll only get better and better, and they won't get any smarter. So that, in that sense, I have hope.
But we all age. So what kind of aging in relation?
Yet, so I agree that it's a battle throughout the whole lifetime, and it just gets harder and harder as we age. That's certainly true. So there's no magic bullet, and you have to punch and count and expect a counterpunch during the lifetime of the patient. But it's a strategy that we will learn to play better and better, and the cancer will not learn to play it better and better.
I want to follow up. Can't the tomorrow's exposures for people be different than today's, so that the originating events that start cancer are different for tomorrow than today, and so in a sense,
Yeah,
cancer can
they'll
change?
probably be less. They'll probably be less cancerous because we're more aware of carcinogens. So that'll probably make it easier. I'm a utopian optimist rather than a dystopian.
Let me give you an example of cancers getting smarter. Hereditary breast cancers get smarter. They anticipate. So if your great-grandmother had breast cancer and there was a BRCA mutation, each generation gets the cancer earlier. So the cancer some way is getting smarter in terms of more aggressive, more expression. So there are cases with hereditary cancers with anticipation. It happens in ovarian cancer, it happens in breast cancer.
But by what mechanism?
Well, that's what we are asking you to.
I am
It's
very.
proposed to be.
That's a Lamarckian statement that raises
people-
my hackles.
Well, people have looked at telomere lengths, which get shorter, and the telomere lengths can correlate with the cancer. So this is a known phenomena. It's called anticipation. It happens in many cancers that are hereditary, and the cancer is getting smart.
It's also Fragile X and other diseases.
But come on, I mean, that's
yeah,
not cancer
that's-
doing it. It's basically just a correlation, right?
Yeah.
Yeah, I mean, that's sample bias.
Yeah, let me defend Dave, because I have to, I have to agree that there, environmental factors may change, our population genetics will be changed, but the cancer isn't going to pass on its information. But let me tell you, there are exceptions. There are two examples of transmittable tumors.
Yes, all right.
There's the Tasmanian devil.
Tasmanian devil, yes,
And there's
all
the
right.
canine transmissible venereal tumor, which
That's
has been
right.
growing for 1,000 years, passing its information along. But the
right.
point is, that's quite distinct. What David's saying is that you're not going to get like a MRSA bacteria, right? If you figure out how to cure a run-of-the-mill colon cancer today, you should be able to cure it 200 years down the road,
Yes.
because it won't pass any
Thank you.
resistance genes. Now, that may not be, if we start having transmissible tumors, that may be a different case. But there is quite a distinction.
I just want to mention something about what Mina had said about age, right? It's not so much, to me, the aging process itself, although I understand the greater question. It's about the immune system's decline, right? And, Mark Davis has done quite a bit of work here about the immune system decline with age. And the long and the short of it is 50 is the inflection point, right? Things really start going downhill after 50. I'm looking around the room. I'm 53. So, but what's interesting is, though, that you can actually find people who are 70-year-olds who look like they've got 40-year-old immune systems, and 40-year-olds who look like they've got 60 or 70-year-old immune system. We just published a paper in Science Translational Medicine this last week about surgery, and showing that there's blood cell immune system components that basically can predict the outcome for your surgery. So that might also be the case about cancer. There might be an immune declination event that you have that basically enables the tumor.
So thank you for your talk. That leads to my question. I wanted to know, is it possible to monitor different components of immune system and tumor microenvironment with respect to perturbation using with and without drug to predict whether the patient will respond to therapy or not beforehand, and try to find a target which can cure the resistance as well, or pathways to overcome the innate resistance? How do you see this technology exponent that into the clinic?
To me or anybody?
Any of you?
How is it going to get in the clinic? The hope is that, there'll be companion diagnostics and early diagnostics that'll measure the heterogeneity, and the therapy will be tailored around that. That's the vision, but there's a lot of work to do before it gets there.
Yeah, I would agree. The sooner we can get to the point where we can have a standard bit of information about a patient who has a study on 100 or 200 genes, or even whole genome study, that what are the actual allelic frequencies, is there a suggestion of subclonality and so forth. That should be part of the report, in a sense, rather than yes, they have this, or no, they don't, as if it was all one, as if there was a simple answer like that. I think it would be good if we got that into clinical practice. It takes time.
I think there's at least two major steps. One is to show, like Nellie's starting to show, that the more heterogeneous tumors are prognostic and that you might want to treat them differently, right? As opposed to more homogeneous, that might be more curable. And then there's also probably we want to be able just to detect the rare resistant mutations we already know about for helping with drug selection up front. But those have to be done before you would actually change clinical treatment.
Of course, the resistance mutations at very, very low frequency are an extreme problem, and the methodology I was talking about doesn't address those at all. Do you have any take on that?
Well, I would just think that if we can get the histology approach working in a sort of more real-time manner, when I think about the heterogeneity, one of the things that we started to do is put in all the apoptosis markers and even necrotic markers, and you actually find regions in the tumor that you probably shouldn't be even paying any attention to, at least from a phenotypic standpoint. And so being able to correlate across the tumor, not just the markers that you think are interesting or the genes that you think are interesting, but whether or not the piece of the tumor is hypoxic, we can use HIF-A1 or other things to call that out. You might want to say, well, what is the drug doing at the in the oxygen deprived versus basically the normoxic region of the tumor, and would you derive that kind of information from a bulk analysis? And so that's where I think this multiple marker measurement system is going to bring its most immediate efficacy in interpretation of the data.
And I guess I would add that Bob Gatenby has been pioneering ways of doing this radiologically, non-invasively, looking at heterogeneity in the tumors in terms of the different microenvironments and looking at how that predicts response.
One more thing on the rare mutation. So if you want to know whether 1 in a million cells have a rare resistance mutation, then you're really going to you can't pick out individual cells and hope you find one with it. You can't bulk sequence and hope you find it from the bulk sequencing data. You're going to have to focus in on that question with a technology that's appropriate to answer that question. And in many cases, we may end up with it for practical clinical purposes with essentially having a lot of statistics and having studied many cancers of the same type and having essentially the information that, well, probably there is one, right? Or if there isn't one now, there will be one, right? Because we know patterns of resistance, and we can expect that these very low frequency events either exist or will happen and then can be amplified. In reason about a class of low frequency events from statistics rather than precise measurement in regular routine clinical practice. I'm not arguing that we shouldn't go out and measure these things, but it's going to require technology that's different from what we've been talking about.
Yeah, and I would argue that measuring heterogeneity is probably a good proxy for the probability that there's a rare mutation, a resistant mutation there, even if you don't see it specifically. Yeah.
Hello, I have a more technical question regarding RNA sequencing, looking at the transcriptome. Gary, I understand from your talk that you're looking at a small section, and you said basically you're working on something you can go to the single cell or single molecule level. So my question is, could you combine both, like single cell sequencing mRNA with that approach, and what could be the advantage, like normalizing the data and more specific? Because one challenge in mRNA sequencing is the normalization. We over-amplify specific RNA species versus some which are really interesting, and we can look at them right now. because you really need to be deep sequencing. And my question is there a possibility to combine those two to actually normalize the data?
Well, so if I were to do that, and I had, all the NIH's resources at my disposal, what I would do is, remember I showed you the 50 slices? So we could shear off a quarter of the cell, and the remaining amount, and of course then we have the spatial information. The remaining amount of the cell, to the extent that the messenger RNA is in the other half of the cell equally as to what it was that we saw expressed, could be then handed off to single-cell sequencing. So yes, it's possible to do that.
Okay, so a couple of you are experts in single-cell measurements, and some of the issues that I've had with interpreting single-cell data is that most of these assays are destructive, right? So you're always taking a sample at certain snapshots in a population's history, and those measurements, if you're really lucky, you'll get the case that Steve was talking about, where you have nested changes in the population. But if you're not, you could have alternative hypotheses where cells are maybe transitioning between different states, and you're never going to capture that information, right? So to me, that motivates going towards live single-cell measurements. So I'm interested in turning the conversation towards where you would see your methods being complemented with methods in the future that might be able to track populations through time with single-cell precision.
Yeah, so, very powerful technology for doing this in live cells is fluorescent reporter proteins, and the drawback is just there's a finite number of colors you can look at. And so, when you narrow in on a question, I think it's pretty straightforward to do the live cell experiment. And when you want to do the global measurements, I mean, that's usually what focuses you, the narrow thing. So they kind of work hand in hand that way.
Yeah, I think another important thing that we haven't really talked much about is sort of the type of pathway analysis that a lot of people are looking at. I mean, there is some encouragement that, in fact, there's only a limited set of pathways that a cell can use to overcome metabolic problems, genomic problems. And so it is encouraging in the sense that when you say, David, that there are these rare mutations that are out there, well, if they're in a significant mutation, they probably affect one of a fairly small number of pathways, right? And so that, I think, is the next level sort of integrating this to analyzing these individual pathways. And, once we've discovered all of the pathways that we need to be looking at, right, then presumably at some point we'll reach a state where we have those pathways, we understand those pathways, the genes that are in those pathways, and then the ways that the tumor can then become a tumor, right, by mutating genes in those pathways. So I guess that brings us back to the tumor heterogeneity idea that there are critical pathways. For example, people have proposed that glutamine metabolism is important for melanomas. Well, there are certain genes that are mutated that affect glutamine metabolism. And so perhaps, we just need to try to integrate more of that into our thinking. And not only that, look at examples from melanoma, which I'm most familiar with, where in fact you have very distinct types of melanomas. You have BRAF positive, you have BRAF negative. Well, in a sense, the BRAF negative have NRAS mutations, some of them have RAC1 mutations. Maybe we're also looking at it in the wrong way. We're looking at melanoma as a single disease rather than a disease that has a mutation in the RAF-ERK pathway as opposed to cells that have mutations in RAS pathways, and looking across cancers, right, rather than simply treating these individual cancers as the same, because they're clearly not, right, and they're very distinct. I mean, BRAF mutants tumors never have NRAS mutations. So they're really like a different tumor, but those pathways, obviously RAS is common to many cancers. So it's really more pathway analysis, I think, that we need to be thinking about.
I totally agree. I mean, I think you view this as a stochastic game between the cancer and the host, and when we see a patient come into the clinic, several moves have already been made in that game. And in fact, the host has already counter-moved, especially with an immune system, right? So we really understand, we have to understand the state of the battle that has already occurred between the patient's cancer and the patient's immune system. Many, many moves have been made already in that battle, and that limits the number of strategies that are available to the cancer and suggests strategies for the treating physician. So to outline those things is great. And we don't want to, a second point I do agree as well, and we don't want to outline this in terms of billions of possible mutations. We want to group these into higher level strategies, like, well, the cancer could respond by upping this pathway. And we have plenty of statistical evidence because, you know, we've seen all these going to BRAF, right? We've seen all these cases where this alternative pathway is now activated, and that is the source of resistance. So, and if you have some idea of the probabilities of this for that particular patient, given the moves that cancer has already been made, then that would select your approach. And if you're doing immunotherapy, if you can measure the vulnerabilities of the cancer in terms of how you would beef up the immune system to exploit those vulnerabilities, then all the better. That's great.
That's true. I mean, the cancer is also inhibiting the immune system.
Right.
So it's those inhibitory molecules that's another signature of many of these cancers, obviously. So, but again, there's only certain pathways that are clearly potent. Maybe the PD-1 pathway is more potent than other pathways, and that's why people are seeing responses in these cancers. But clearly, there's a limited number of molecules that a tumor cell can employ to inactivate the immune system, as well as to deal with genetic problems, metabolism problems. You know, there's a finite universe. We're not talking about something.
Absolutely. No, the cancer is made the move, and now it's dependent on its own activation of the immune system through PD-1 or CTLA-4. Then you block that. Obviously we love the clinical results from that are stunning. It's a great counter move.
Right.
I would add to that certainly there have been these moves, but in general, we don't have a good grip on what have been those selective pressures that have led to those moves. What were the limiting resources at different stages of its growth? What were the things that were really affecting its death and proliferation? And so we don't have a great characterization of what those moves have been in response to, or that is, those moves.
I want to cast a pessimistic note on this particular point. So people will make exactly the same arguments for antibiotic resistance. So you see particular pathways in resistance to antibiotics, and therefore, if you can somehow prevent those pathways, that one will be done. But certainly, there's a lot of evidence that that's not the case. That if you try to block those ones, there are under somewhat different selective pressures, including lower antibiotics and so on. You'll get many different types of mutations coming up in different pathways that are very hard to sort of explain what they're doing. Now, of course, with the case of cancer, I mean, coming back to David, if one could push it off, and so these are sufficiently rare to happen, or selected on sufficiently weakly that they grow sufficiently slowly that we wouldn't die of those cancers until we were age 150, then maybe we're okay, at least until our ages start going up even further. But that presumably comes back to the aging question. But I think the sort of idea that there are only a small number of ways, I guess I'm somewhat troubled with the evolutionary processes, with the rareness of the mutations, stochastic, but there's a whole spectrum of how rare they are, and duplications and so on, and also the selective pressures. Things are slightly more strongly selected than others. One may end up in certain situations, but always sees those, but that doesn't mean there aren't other ones lurking behind as possibilities that may only be slightly more weakly selected and will take a few more years to develop and so on. So
it's Jurassic Park. Life will find a way.
Yeah. Even though it doesn't get cleverer from generation to generation, I guess.
I think maybe the answer then is to stop thinking about the pathways themselves, but to think about the process by which the pathway mutations occur, right? I mean, so one of the things that people have seen in stochasticity of tumor, or stochasticity of expression, is that cells, even just sitting there, go through multiple states. And there's a lot of focus on mutations in epigenetics. So why are all these mutations coming up in epigenetics? I think that the tumor is using it as a diversity generator. It's using it as an opportunity generator, as it's sampling multiple spaces. So maybe part of the answer is to freeze the generator of diversity, right? Rather than going after the particular mutation and playing, whack-a-mole, why not go after something which is generating the diversity?
Reduce the evolvability of the tumor.
Well, JQ1 is any of a number of different epigenetic modifiers that people are working on, right? I mean, one of the reasons why I put up that epigenetic thing there, because that's what I think is going to be the next level to go after. I want to know what the molecule by molecule, gene by gene, nucleotide by nucleotide arrangement is in cells, right? And then ask the question, what's going on in the diversity of multiple cells in a population? That would probably give us some clues as to what molecules we should be going after.
So what I wanted to ask Steve and
Yeah, move closer to the microphone.
sorry. What I wanted to ask Steve and Gary in particular is how we reduce this to a clinically treatable level now. So we just did targeted sequencing on a woman who had ER-positive, HER2-negative breast cancer in 2008. She's had nothing else except for about 3 bouts of hormonal therapy. And we did targeted sequencing. She's got an FGFR1 amplification, a GNAS and a KRAS amplification, JAK2 mutation, Aurora kinase A amplification, MYC amplification, TET2 mutation, MAP3K1 mutation, and a MIS3 amplification. So all we want to do is basically subdivide these into which are the key subclonal populations. We're trying to treat this woman. So we want to just get this down into 5 or 6 key subclonal populations to which we could give treatments. And then we know that the tumor is going to rebound, so we'd love to follow this in a single cell level, if possible, or whatever level, in CTCs or circulating nucleic acids. How reducible is this to real time now? In other words, how much can we add this on to the current kind of foundation medicine sequencing we're using?
I think it's early days. All right, you've got to be blunt about that. Yeah. And these technologies are very much on the cutting edge. We're just figuring out how to use them, and so there's a path to that. But I don't think any of us are saying right now to go take them into the clinic and it's ready to be used. But it's setting us up to get the knowledge to use them, and I think as a community we're on a path.
Yeah. What I would say on this is we're not looking for 100% precision on this. We're just looking at putting some order into the subclonal populations.
Well, I would hope that to the extent that whole genome sequencing becomes useful in the clinic and often used in the clinic, and everybody's predicting how many years this will be, but at some point I would say there will be whole genome sequencing in the clinic, then we clearly will be able to detect large subclones. So if you had a subclone at 30% and a subclone at 20%, these diagrams are easily computed from whole genome sequencing. So so some clonal analysis, at least for the larger ones, will come along with whole genome sequencing, and that's because you have so much information across the whole genome that you can start to get statistical confidence in calling a subclonal structure at some crude level. So that's good news. So that can happen.
I'll say something else, alluding to something Gary just brought up, that once you're already sick, the diagnostics have limited value because there's not that many treatment options right now. And a big part of this is predicated on the hope that there are going to be sort of a new suite of therapies, whether it's epigenetic drugs or others. But even just forgetting about the heterogeneity part of it and doing tumor sequencing as it exists today, some people it helps, but there's a lot of people it doesn't help. There's mutations. We know that, they have, effects, but there's no way to treat those specific mutations. That's not an uncommon result for someone who gets a foundation medicine test today. And so when you're at the point of being sick and doing the diagnostic at that point, the value is strongly coupled to the availability of therapies.
As per my earlier question to Nelly, we are taking this new adjunct, so we do think there's hope.
Right.
So one of the things that I've been interested in here is to ask the question about, I'm going to change my mind. I won't say that because I think it's wrong.
Good thing you caught it.
Yes, I caught it at the time. I was thinking ahead.
I'm never afraid to say something wrong. I was struck by Gary's remark saying that cancers just can't evolve in any direction. They're bound by certain constraints, and also the discussion of pathways. Are we looking at a problem where everyone's heterogeneity is different, or are there going to be patterns of heterogeneity in people where we can think about putting together research databases of heterogeneity?
Well, this relates to the question that the woman asked me about merging genetics with the phenotype, right? And so, you know, we've said it. There are a limited number of phenotypes available for any given cancer. So one of the things that we're doing is now that we have all of the phenotypes, or we can mark them out into these different clusters, sort those, and ask what is the genetics in 50 different tumors, patients, of the cells that sit in that cluster, right? There's probably going to be multiple genetics that are all contributing to a common phenotype, right? You're not treating the genetics, you're treating the phenotype. So the extent to which we can understand what the limited phenotypes are, it doesn't matter what the genetic solution is, right? We're treating the phenotype.
Right, although if you're binding to a particular pocket,
Yes.
then
Then you're, that's what I mean. You have to, there's somewhere at the apex of all of that, there's almost a housekeeping function, right, that they're all using that is going to be the limiter.
Can I pick up on that? That last point was exactly what I was hoping you guys would talk a little bit more about. So you guys have delineated very powerful technologies in elucidating the landscape underlying a lot of these cancers. But ultimately, that landscape changing over time and in response to treatment is the things that are most proximal towards getting an outcome for cancer cases, for those of us treating it. So some of the approaches you guys have outlined are terminal approaches, CyTOF, whether or not you put it on a Fluidigm chip and take a look, and whether or not you look at that sequentially over time. Can you guys talk a little bit about approaches that you guys are employing now, or that you see would be amenable to these new technologies you're developing that would let us get closer to not so much phenotype in terms of protein expression, but phenotype apropos of the first talk this morning of populations that support or maintain or establish malignancy?
He's looking at me.
Yeah, I'm trying to think about this one.
Go ahead.
So I'm just, maybe I could push you a little bit to clarify your question a bit. What aspect of the terminal measurement is it that you don't like?
So there isn't necessarily a relationship of the landscape to a functional outcome for a person, right? We don't have the link. If everything is stereotyped, if the evolution is all simple of A goes to B goes to C goes to D, then hey, it's great if you figure out how much A, B, C, and D there are. But if the evolution, sort of apropos of the antibiotic kind of resistance evolution, if there are dynamic pathways that change over time, taking a static measurement of the landscape of a cancer even at a given time, hey, molecular diagnostics is low-hanging fruit, and that's going to get us somewhere, but that's not going to get us to the point of how is a given cancer going to respond to a given treatment. How do we get closer to that answer using the very powerful diagnostics you guys have talked about?
Well, you could come in, you could do an allele state diagram, you could see the patient has 4 measurable subclones, you could treat them, and you could resequence them, and 3 of them have gone away and the other one has expanded. That is potentially useful information. That's certainly doable within today's technology. It's not outrageous. It's not cheap. You have to sequence them again. So you would have to sequence the patient multiple times during the course of therapy.
Right, and that's been done to a certain extent. But what that has also shown, the AML data is probably some of the best examples of it, is there's significant case-to-case variation in the way that each of those cases evolve.
Right.
So there isn't a stereotyped evolution in response to even the same sort of treatment.
It's all statistics. So we had to, once we do this on a scale of hundreds of thousands, then you start to see patterns.
I'd actually push you to say maybe it's not so much statistics as it is biology. And so in that context, that are there ways, for example, that you see CyTOF or immunophenotyping or using a single cell coupled DNA and RNA sequencing to relate to, do you see that being, if we do cohorts prospectively over time, is that something that would have actually additional benefit from the way of doing retrospective analysis of the cases that already exist? Are there ways that you can plug those into things like, colony competition assays or growth assays? Do you think those kind of approaches would yield any benefit on top of the kind of diagnostics that you guys are already talking about?
Well, I guess I come back to Gary's earlier point, which I'll try to restate. Cancer, while it's a very idiosyncratic disease, is not infinitely malleable, right? I mean, there are constraints on what parts of a given tumor are going to do that's dictated by the basic biology of the cell types. And the hope is that, you're going to be able to find that middle ground, right, where there's some ability to characterize the clones according to a recognizable phenotype that, maybe it's not the same genes mutated from patient to patient, but they'll be within a related pathway and establish a kind of canonical type that one will be able to generalize around. And that's a hypothesis, but not an unreasonable one, I think, looking at the balance of what we know about cancer today.
I think part of what you're asking is around the spatial organization of the niches within the tumor, right? I mean, as Cornelia showed this morning, that there are lots of places where two tumors two clones in sufficiently proximal, region where they can communicate with each other is a beneficial thing for both of those pieces of the tumor. So, with CyTOF, we can see those kinds of events with a correlation analysis, right? You'll see that basically when one cell population is up, the other is down. We've done this lots of times with the immune system already. So that would be one way to do it. But then if you actually had the spatial organization via some kind of histologic approach, let's say that we're, the MIBI thing is still, in the future, but let's say that we did some limited histological, so we did the CyTOF, found what the relevant markers were, we could go back to traditional histology and ask the question, are these two cells, how close do these things need to be to each other? Right? I mean, those are the kinds of questions that you're asking, right? And if there is a dependency of their closeness, maybe knocking off one of the two or cutting off their communications channel, IL-11 or other things, that would be the key. So, I mean, I think it's going to be about collecting the information. And I don't think that phenotype is different than genetics, right? Genetics is phenotype. I mean, it's just, it's just a way for us to keep, out of each other's hair for the time being. But, I mean, I think they are coming together and they should be thought of as the same.
I'd like to try to answer Sid's question in a way that I thought Gary would. Gary made the point that if you interrogate the audience, you can find out something about them by the way they answer. And I think you're asking the question, how do we predict what's going to happen when we try to treat a patient? So if we can ask the cells, what will they do when I expose them to this drug? How will they behave? How will they respond? That gives you information that's not a terminal analysis. That's a functional analysis. So I think that the approach that Gary articulated about going inside the cell and looking at the signaling pathways in response to a perturbation, that's not a frozen time in analysis. That's a forward analysis that predicts something.
So I buy that to a certain extent. I mean, I think the analogy with antibiotic resistance is actually a great one in that regard, is that even the response to inhibitors of signaling, so targeted therapy, the way that a given case evolves in response to that targeted therapy may vary. And so to the extent that that static picture at the point that you take it establishes your landscape, that...
Well, you have to resample. You have to resample over the course of the patient's course of therapy and ask the how will you respond question now? How will you respond tomorrow? How will you respond after you've seen that drug to the next drug I'm going to try? Okay. So there are dynamic ways of using the current technology. But I'd like to make another point, and that is people are asking for how to predict the future when we don't know what the future is. And the technologies, as we've seen this morning, are going to continually change. They're going to get better. They're going to get sharper. They're going to get more precise. And we have the outcomes of the patients that we treat. The trick is to collect the informative samples. If we have repositories of informative outcomes, as the technology changes, we can keep going back to those informative samples and ask, could this new technology predict? And so that's a pitch for collecting as we go the informative materials that allow us to test our new technologies to see whether they're any good at predicting.
So I want to just...
Hear, hear.
Yeah, So, you know, I didn't show the 20 or so other ovarian cancer tumors that we had worked on because the manner in which they had been collected basically rendered them anywhere from 30% to 60% viable and lots of other problems. When you go back to the history and look at them, some of them sat on the you know, in the operating room. Some of them went through, you know, basically varied people's hands. And I have to say that I'm disappointed. Obviously, a lot of people here are. I mean, and Bev and I have had this discussion about how do we bring the kind of precision that we need. So we've been working with a company in Divimed in Germany, and they literally have a nurse in the suite as the surgery is being done. The moment the tumor is clamped from the blood vessels, she writes that down so you know how hypoxic it might be and how long it's been. And then they immediately move. Everything is done within 3 to 4 hours to frozen material. When we did that on all of those samples there, they were 90% viable, right? And so that kind of reproducibility is what we need. And to be honest, I'm having trouble here at Stanford getting that enacted. People, the surgeons just don't care. Next, you know, next.
Well, collecting is one thing we can do. Collecting in relating it to what happens to that person under that therapy that we can go back to when the technology improves. That's one thing we can do. But I think I'd also make a pitch for not trying to change the practice of medicine for the sake of the technology, but rather to bring the technology to the practice of medicine. I think the MIBI technology has that potential. We collect these things now. We have repositories of these blocks now. We have all these patient outcomes that we know. And if the technology can come to the paraffin-embedded blocks that we currently collect and currently know the outcomes of, that's an example of bringing a technology to the current practice of medicine, rather than hoping someone's going to change the way they process samples or collect samples in the future.
I would add on to that. One of our frustrations has been that many of these tissue banks only put one sample from the tumor into the bank. And if you want to look at these regional variations and heterogeneity across space, then you're out of luck. And so there is an issue of any given study in the future may have requirements that we can't, it's hard to anticipate what, you know, how to store the tissue for sampling.
Hi, Gary, I had another question for you because you're now making a pitch for looking at the epigenetics. And I guess there's something I've never really understood because, of course, epigenetics controls gene expression, but obviously every time you get an answer, it just leads to another question, right? I mean, something had to cause the epigenetic changes as well. So, I mean, does it still come back to, do you think, the mutations in the genetics that cause that, or is there some other cause of that? I mean, how do we think about epigenetic changes, or how do you think about epigenetic changes and what they mean?
Well, I start by thinking of evolution as not the direction. I think of it as a devolution, right? At the heart of every cell is sort of a furnace of the desire to divide, right? The most primitive original cell, right? And facing stress, the cell enacts programs, right? The so-called SOS response, right? You can see it as far back as bacteria. So, you know, people talk about cancer as a movement forward. You know, I think evolution has all been about continuing evolution of control, right, and controlling the systems. So the reason why I think cancer is so easy, because there's so many different ways you can chip away at that control to find the furnace in the center. Right, so the first level that you can do it is dysregulation of epigenetics. You can basically start to force the cell into a plasticity state, an SOS response, let's say. That's my thinking. I have zero proof of it. Right. But that basically is a dysregulation of the epigenetics that then enables the mutations to occur more readily. Right, and if you get the right sets of mutations, you further, it's got to be about the generator of diversity, to my thinking, right? It's got to be about not hoping that you get 2 mutations in a cell, but setting up conditions where mutations can occur. And to me, that's the epigenetics. The epigenetics is the first level. I don't need to mutate things to dysregulate a cell's biology such that more mutations occur, right? I don't need to make mutations. I can dysregulate and enable it.
So you think that somehow there's a stochastic process that a cell will occasionally dysregulate at the epigenetic level? Let's say certain loci will be demethylated or histone acetylations will change. And that's maybe a random stochastic thing similar to what a mutation, right? And then that initiates
the cell is going to be going through states. And if you can push it outside of those states, that might be a place where things that normally control no longer have the ability to control. Right, so it's again the idea of searching for opportunities, right? The the more you can allow for that dysregulation, the more opportunities are available for the cell, as long as they don't walk off a metabolic cliff.
Let's give Charlie and then Irv the last two questions, and since Irv will be the last person, he'll be the one threatening his own time during the lunchtime speech, so he'll have to keep it short. Charlie.
I'd just like to follow that point on that Paul and Gary made, actually, because in renal cancer, this is beautifully illustrated by mutations in PBRM1 and SETD2. So SETD2 is an H3K36 trimethylase. PBRM1 is part of the SWI-SNF complex. And both of them actually initiate, we think, intratumor heterogeneity from the literature and from work we're doing at the moment. So PBRM1 has been published by Jessica Downs at MRC Genome Centre in Sussex. She's shown that loss of PBRM1 generates chromosomal instability and actually inhibits transcription at the point of DNA damage. So we've got double-strand breaks. PBRM1 functions to block transcription, so you essentially prevent R loops from forming, which are a source of genome instability. So presumably, PBRM1 is inactivated in renal cancer to generate chromosomal instability and diversity. So it's almost as if these tumors have to generate diversity, and PBRM1 loss of function is one mechanism of doing that. Similarly, SETD2 is often lost around about the time the tumor starts to branch. And SETD2, as I said, is an H3K36 trimethylase, and it functions in homologous recombination repair, as well as preventing replication stress at the DNA replication fork by enabling nucleosome remodeling distal to the replication fork. So there are two examples where epigenetic modifiers generate diversity within a tumor in kidney cancer. It's a pretty interesting area, I think.
Thank
you.
So I want to go back to what Gary just said. We think of cells as if they're absolutely static in their gene expression for life, but in fact they aren't. Let me give you an example. Hematopoietic stem cells reside in the bone marrow and respond to a particular environment. Every once in a while, normally, and deliberately when doctors do it, they mobilize out of the bone marrow to the blood. Before they go into the blood, they change their epigenome and what happens. Then they go someplace, specifically to another bone marrow site. They have to cross a very hostile field of cells, and they are prepared to recognize the site and cross it, and then they go back to the normal epigenetic state. Same with any lymphocyte that homes throughout the body. Any lymphoma derived from the lymphocytes that migrate. When we did a clonal analysis of both gut epithelium and germline development, even though you may have a mixture of cells that start the gut when you make the tube, or start the germline in the developing testis, something happens and a clonal progenitor has to move from one stem cell niche to another to take over that niche. And it happens over and over and over again. A resting stem cell in the gut or germline stem cell in the testis has a different gene expression when it's resting or proliferating than when it has to migrate to another niche to take over that niche. So this is a plea for spending cancer money, National Cancer Institute money, for understanding the biology of these different systems. Because we have cancers of those cells that have a biology that's inherently plastic by epigenetic change. And they can go back again, so it's not a permanent epigenetic change, although many cancer epigenetic changes, you don't go back.
Any comment, or I want to make that the last word?
Yeah.
Yes. I think we all agree with you.
I agree.
Yes.
Yes, here, here. All right, we will break for lunch then.